We are learning about genetics. And remember, this is going to help explain how you got some of your traits, your physical appearance. And so we've started learning these words. We took these notes a little bit before on genotype and phenotype. And we just want to review these words before we go on because they are really important. Your phenotype is your physical appearance. And so when we look at the ability to roll your tongue, or have a hitchhiker's thumb, or have unattached earlobes, those are my phenotypes, or my physical appearances. But my DNA is actually coding for me to have those traits. So when I can roll my tongue, I'm actually showing the dominant phenotype. I have a dominant genotype, but I don't know if I'm homozygous dominant or if I'm heterozygous. And so we have these two words to describe because homozygous, like big A, still gives you the dominant appearance, but big A, little a also gives you the dominant appearance. But if you're big A, little a, and you have a recessive allele, you can pass that on to your offspring. So this is why it's so important to understand the word heterozygous, because it means that you're hiding a recessive allele. So we have these words that we want to practice a lot: homozygous and heterozygous, genotype and phenotype. What we're going to see is that there are three main principles or rules. The first rule is that dominant alleles can mask or hide recessive alleles. So let's look at this one, the heterozygous. When we look at heterozygous, we can actually see that heterozygous is where I have two different alleles, a dominant allele and a recessive allele. But I do show the dominant version of the trait. So for example, being able to roll your tongue. If I can roll my tongue, I might be heterozygous or I might be homozygous. If I cannot roll my tongue, then I must be homozygous recessive. So what this rule is saying is that if you have recessive, we know that you are homozygous recessive. If you have the recessive appearance, then that means that your genotype is all recessive. But if you have the dominant physical appearance, it could be because you have two dominant alleles, or it could be that you have a dominant and a recessive, because just one dominant allele can hide or mask the others. So that's Mendel's first principle. And so when we're solving these problems, we actually have to practice remembering that dominant alleles can hide or mask recessives. So if I show up dominant, I might be hiding a recessive allele. The next rule is the principle of segregation. If you know the word segregation, like from history class, you know that segregate means to separate. And so segregation means separate. What that means is that during meiosis, the homologous pairs will separate. Remember, inside of your cells, your chromosomes show up in sets of two. You got one from your mom and one from your dad. We copy them and then we divide them so that each family only has one of each type. And that is Mendel's principle of segregation, that you actually only inherit half of your genes from each parent. This is going to be really important when we start doing what's called a Punnett square. Punnett squares are going to use all of Mendel's principles at the same time. So we have the principle of dominance and the principle of segregation. You might want to make some flashcards to practice these. But as you can see, each parent only donates half. The third principle is the principle of independent assortment. Independent assortment means that you get your traits independently of each other. What this means is that just because I have brown hair doesn't mean that you have to have brown eyes, because those traits might be on different chromosomes. So for example, if the trait for brown eyes is on the big chromosome and the trait for brown hair is on the little chromosome, I might inherit them separately. And so independent assortment tells us that genes on separate chromosomes, like the eye color gene and the hair color gene, if they're on separate chromosomes, they are passed on independently from one another. So these are Mendel's 3 rules. This is what Mendel got from studying the pigments, that some traits are dominant, some alleles are dominant, and they hide other alleles. That you only get half of your DNA from each parent, and that your traits can assort or mix independently. And so a man named Reginald Punnett actually developed a way to track genetics. He created something called the Punnett Square. And the Punnett Square is the way for us to track the genes that we're inheriting and predict which versions you might pass on to your children. So Punnett Squares, oh no. Sorry, getting some glitchiness here on the computer. Punnett Squares are used to predict we want to add in the word probability of certain outcomes between the gametes. The sides of the Punnett Square are labeled with the genotype from each parent, and we cross one trait at a time. So if you look at the pictures that you have, you can see that we put the father along the top and the mother along the side. Now the father, if you look here, was probably big T, little t. That was his genotype. And the mother was big T, little t. The father must have been dominant in his physical appearance because he had a dominant allele. And the mother was dominant in her appearance because she had a dominant allele. However, when they went to have offspring, meiosis separated the 2 alleles, so that one sperm passed on the dominant allele, but a different sperm was carrying the recessive allele. Same with the mother. One egg had the dominant allele, a different egg had the recessive allele. Remember, this is Mendel's principle of segregation, that during meiosis, they separate the 2 alleles so that you only pass on one. Now, if this sperm from the father were to fertilize this egg from the mother, the offspring would be homozygous dominant. Now that's not what either of the parents were, but it's possible because the dad had the dominant allele, the mom had the dominant allele, and so this offspring inherited 2 dominant alleles. Now if this sperm and this egg came together, we'd get a heterozygote. They'd still be dominant. All of these would be the dominant phenotype because they have the dominant allele. But this one would actually be recessive. And that's what's so interesting. We have 2 parents who both had the dominant physical appearance, but they each passed on a recessive allele, and they have a child who has the recessive appearance. And so the Punnett Square is actually brilliant because it's explaining this idea of segregation or separating the alleles, fertilization, and then the physical appearance. By looking at these possibilities, I can determine I can predict the physical appearance. Now, if this mom is pregnant, I don't know which of these 4 she's going to have, but I can make a prediction. I can say that 1 out of 4 will be recessive, and that's going to be 25%. So 25% of their kids will show recessive. 75% will show dominant. So when we look at a Punnett square, we can make predictions about what the offspring will be. Another important point is every new event, every new birth, starts the Punnett square all over again. So just because we have one baby who's recessive doesn't mean that no more will be recessive. Each fertilization starts over with the probability. And so what you need to be able to do with a Punnett square is you need to be able to take a genotype and predict the genotypes and phenotypes of the offspring. You can also do what's called a dihybrid cross. So let's say we wanted to know the chance of rolling your tongue and having unattached earlobes. Those are 2 traits, and I can predict what would happen if I had both. But you see, we have to actually use a very different setup because we have to predict how often they would combine in the gametes or the sex cells. So we might have a sex cell that has 2 dominant or a sex cell that has 2 recessives. So this is actually where we're going to stop with the notes for tonight. It is actually really important that you, oh no, we're not. We have one more to solve. Oh good, we have a practice problem. Okay. My little toolbar keeps going away every time I click to the next slide. So here's an example. Huntington's disease breaks down the nervous system. It is a dominant trait. If a heterozygous woman, now remember, the only way to be heterozygous is to have 2 different alleles. In this case, a dominant and a recessive. So here's the mother. She's heterozygous. If she marries a man without Huntington's, what is the chance their children will have the disease? Well, here's where we have to be careful. If having Huntington's is dominant, that means that big H has Huntington's, big H has Huntington's, but little h does not have Huntington's. So the man without Huntington's, we know, has to be little h. So you can see that the mother's genotype was separated at the top, the father's is separated down the side, and then I can combine them. What if this egg was fertilized by this sperm? What if this egg was fertilized by this sperm? And so we each time are representing a fertilization event. Then we answer the question, what is the chance their kids will have the disease? Remember, it's dominant, so this one is dominant and this one is dominant. That's 2 out of 4. 2 out of 4 is 50% of their children will have the disease. Next it asks for the phenotype ratio. So we're going to write that as how many dominant compared to how many recessive. Now we have to be careful. Ratio means comparing part to part, versus a fraction compares part to whole. So a ratio compares the number that are dominant to the number that are recessive to. And that is our phenotype ratio. But then it wants to know the genotype ratio, comparing part to part to part. Now we have 3 parts, homozygous dominant, heterozygous, and homozygous recessive. So if you'll please fill in how many are each of those genotypes, keeping in mind you can say 0.